Hello, I'm Tim Weaving, a PhD student at University College London, and I'm pleased to welcome you all to the 21st instalment in Comp Biomed's e-seminar series in collaboration with the Virtual Physiological Human Institute, or VPHI. The talk today will be given by Ivan Benemerito of the Insignia Group at the University of Sheffield, which he has been a part of since 2015. Ivan has a background in mathematics, and his PhD was concerned with modelling the ankle joint. He joined Comp Biomed in 2018 with the purpose of developing methodologies for improving the diagnosis and treatment of ischemic stroke via a combination of deterministic and statistical modelling. In today's talk, Ivan will give us an introduction to Gaussian Process Emulators, or GPEs, in conjunction with sensitivity analysis. Furthermore, he will also present two applications of GPEs in the cardiovascular and musculoskeletal domains. Now, before we begin, I'd like to briefly remind you that the session is being recorded and will be available on the Comp Biomed website and YouTube channel shortly. So please do use this to go back over parts of today's talk and others in the series, and perhaps even share these e-seminars with your friends and colleagues. Now, there will be a Q&A session at the very end of the talk, so please save all of your questions until then. Once the talk is finished, you can go ahead and post your questions in the questions tab, and I will then read them out to Ivan to keep the session running smoothly. Now, that's everything from me. I very much hope that you enjoyed today's talk, and without further ado, I'm going to hand over to you, Ivan.
Thank you, Tim, for the introduction, and good afternoon, everybody. So, as Tim said, today I'm going to talk to you about the Gaussian Process Emulators, and I will first give an introduction on what they are and how to use them, and then I will present two applications. So first of all, I want to start with the rationale behind this talk and explain why do we care about Gaussian process emulators. And to detail, I will start looking at the things from their endpoint. So we all know that computational models are increasingly used in all domains of science and technology, and it can be for research or commercial applications. And in case of commercial applications, the regulatory bodies require that the computational model is credible and it can be used safely. But of course, also in research, we want to make sure that our models are reliable and credible. And the credibility of computational model is typically assessed in stages, and these stages are verification, validation, and uncertainty quantification, or VVUQ for short. And verification is the step where you check that your computational model correctly solves the equations that it's supposed to be solving, while the validation is the phase during which you make sure that the computational model correctly describes the physical phenomenon that it's supposed to be describing. And finally, there is the uncertainty quantification, which is particularly important because practically always the input parameters of a model are affected by uncertainties, and we cannot know them exactly. And the uncertainty quantification measures how the input uncertainty propagates and affects the output, and also gives way to rank the input parameters according to their importance and their effect on the output. The very important question is, how can we quantify the importance of the input parameters? And there are a variety of techniques for doing this, and the one I will be briefly talking about today is Sobol sensitivity analysis. So imagine that you have a computational model with uncertain inputs, and these inputs have an associative statistical distribution. Now you run your computational model and compute the corresponding outputs, and they will also have an associative statistical distribution, which you can characterize, for example, using the mean and variance of this distribution. Now, what Sobol sensitivity analysis tells you is how you can decompose the variance of the output, and which you can think of this variable v as the variance of the output, And you can decompose it into contribution from the variance of single input, or by the contribution of the variance where multiple inputs are modified at the same time. So with Sobol sensitivity analysis, you can say that a particular input is responsible for X% of the variance of the output, and while combinations of input are responsible for another percentage. And in this way, you are able to rank the inputs and identify which ones are responsible for the largest variation in the behavior of the model. And these outputs, these inputs are actually the ones that you want to control, and whose uncertainty you have to quantify with more accuracy, because in this way you will be able to trust more the outcome of your computational model. Now, to estimate this input-output dependency, it is fundamental that you properly explore the input parameter space of your computational model. And to do this, typically you have to perform several evaluations of the model. And the rule of thumb is that if you have a model with n input parameters, typically you need in the order of 1,000 times n model evaluation for computing this input-output relationship and ranking. And now this can take a long time, even if the computational model is relatively fast to run, but becomes practically, almost impossible for models that are more computationally demanding. And in this case, a faster solution is needed for proper exploration of the parameter space. And a solution to this problem, a faster solution, is the use of statistical emulators. Now, statistical emulators are a statistical approximation of a simulator. And when I say simulator, I mean the computational model whose behavior you want to investigate. Now, imagine that this gray region here is some sort of space of parameters. And the function f here represents the simulator. So this is actually applying f to the input space is actually performing the simulation with the computer. And what f does, it maps the input parameter space x into some output space y. When you develop an emulator, what you are really doing is developing an approximation of the simulator. In this case, is this function f tilde here. And the action of f tilde on the input parameter space produces an output space y tilde, which is similar to the target output space, but it's not exactly the same. And the degree of resemblance of these two spaces can be measured using some criteria that have to be decided at the beginning, and you can also evaluate afterwards. Now, I said that a simulator is a statistical approximation. That an emulator is a statistical approximation of a simulator. But what does it mean exactly? It means that for each point of the input parameter space, you actually have a distribution of functions that are possible candidates for working as an emulator. So in each point here, you have an infinite number of functions, and what you need is a criterion. It's a criterion for discriminating among these infinite possibilities and pick one function that emulates f in a satisfactory way. Now, one of the requirements for the statistical emulator is that the average of this distribution of function actually behaves as the simulator that you are studying. And another very important thing about the emulator is that when they provide an estimate of the simulator, they also provide an estimate of how likely the mean of this distribution is to be close to the desired function that you're targeting. A very important thing about the statistical emulator is that they work leveraging on Bayes' theorem. Bayes' theorem is a very important theorem in statistics which answers to the question, what is the probability of observing a certain event given the fact that I have previously observed other events? So consider, for example, this square here, and behind the square there is an image that is hidden. At the very beginning, you don't have any idea on what is the image that is hidden behind the tiles because you don't have any information. But as soon, as we start removing tiles, you start to see bits of the images and you start to develop an idea in your mind of what the entire picture could be. And in this case, obviously, it's a picture of Ludwig van Beethoven. And statistical emulators work in essentially in the same way. So they need an ensemble of observations, which is also called the training data set. And from this training data set, the statistical emulator can infer the nature of the system under consideration and the behavior of this function f. And a very important thing about the statistical emulator is that their error they make on the training data set is 0. So when you compute your emulator and you evaluate it on the training points, you have exactly the same behavior as the simulator does. Now, having said this, when do we actually want to use an emulator and what can we use them for? So they can be used for interpolating the input-output mapping of a complex simulator. And this can be done by using only a limited number of simulator runs. Or they can be used for doing forward propagation of uncertainties. Now, the first case is, for example, when we know the input parameter space of the model, and we know what are the values that the parameters assume, but we, for example, know the value of the simulator only in a limited number of points, for example, P1, P2, and P3 in this case. Now imagine that you know the function, the value of f on P1, P2, and P3, but you need the value, you need to evaluate your model in P4, but for whatever reason, it's not possible for you to run the simulation. So in this case, what could be done is to train an emulator on these P1, P2, and P3 points, and you then use the emulator to provide an estimate of the behavior of the system in P4. And what is very interesting about this approach is that if the emulator is trained sufficiently well, you can actually use the emulator to generate the large number of points that are needed for sensitivity analysis and for parameter ranking. And this application is the one that I will describe later, and we will focus on this throughout the rest of the presentation. But I would also like to spend a couple of words on the forward propagation of uncertainties, because as I said, it happens very often that the input parameter space is not exactly known, and you may find yourself in a situation where you only have a statistical distribution of the input parameters. And in this case, what could be done is to train the emulator on the data points that are available, and then use the emulator to compute the output distribution that results from this known input distribution of the parameters. Now, associated with these two applications, there are obviously a number of problems. I mean, number of things that must be taken into account. And for example, one is how do you effectively sample the input parameter space so that the emulator learns how to correctly imitate the behavior of the simulator, and what sampling method should be used, and how many points must be sampled. And there are all these sort of questions whose answer often depend on the specific application that we're dealing with, and we'll not discuss them further here, but it's important to be aware of these issues. Okay, so now let's be a little bit more specific on what the Gaussian Process Emulators, or GPE, are. So being emulator, of course, they are a probability distribution over function space. And as I said before, on each point of the input parameter space, you have an infinite number of possible functions to pick. And the GPE is able to rank them by assigning a probability distribution. And this probability distribution is also refined as more and more observations of the system are performed by feeding the emulator with the training dataset. Now, the Gaussian process emulators are a kernel-based method, which means that you have to define a sort of scalar product into multidimensional space, which is used to compute the input-output approximation. And to do this, you will need to define a specific covariance function and matrix. And we make here the very important assumption that the approximation of the input-output relationship is smooth. This is actually not entirely surprising because the word Gaussian is actually in the name of the method, and the Gaussian advocates the idea of smoothness. With here prior and posterior distribution, what is meant is that before performing an observation of the system, you have the emulator has a certain idea of how the system might behave. And this is given by the probability distribution that is defined on the input parameter space. But then after, I mean, as you train the emulator, this probability distribution changes because it has to accommodate the information that is available on the system. And the probability distribution that you end up afterwards, after the training, is called posterior distribution. This is a quite clear example, actually, of how a Gaussian process emulator works. So imagine that on the x-axis, you have the input parameter space of a computational model. On the y-axis, you have the output, and you want to estimate a function that is defined on this input parameter space. Now, at the very beginning, here, you don't have any information, and you can't pick any function over the others. But as you start adding training points, you see that the uncertainty on the estimate is reduced. And first of all, I mean, you can see that behind two known points, the uncertainty goes literally to zero, and your emulator it gives you a clear estimate of what is the function that you are trying to estimate. And also note how the uncertainty is zero on the training point, and this is one of the requirements of a statistical emulator. Now, a Gaussian process is defined by its mean and covariance, and these are computed after, as I said, after observing how the simulator behaves on the training point. And the covariance is constructed using a kernel function, and the process of training the Gaussian process emulator is actually the process of tuning the parameters which are inside of the kernel in order to minimize the discrepancies between the behavior of the emulator and the behavior of the simulator. And of course, as you can imagine, the discrepancy between this behavior is quantified by a loss function. And this is made quite clear in this example. So we are using here, we're looking here at this emulator with a kernel that is called quadratic exponential kernel. And it's exponential because there is the exponential here, and quadratic because of this scalar product here. And this is essentially a multidimensional Gaussian. And the kernel in this case is this matrix K, which is composed by the product of a rotation matrix R and a scaling matrix lambda. And together, R and lambda define this red ellipse in the input space. So imagine that y1 and y2 are the input parameter space, and you can, using these two matrices, you can define an ellipse with the semi-axes lambda 1 and lambda 2, which come from the matrix lambda, while the orientation of the ellipse is given by the matrix R. And again, at the beginning, you don't have any observation on the system, so there isn't any preferential direction for the ellipse to be rotated or stretched. But if you add and observe, if you observe a data point in the, and then go performing the training, there is this data point here, and what will happen is that the training will modify the parameters inside R and lambda so that you minimize the, you maximize the fit with the training data. Now, this slide shows quite clearly the difference between the Gaussian process emulator and other interpolation techniques, also nonlinear interpolation techniques. So the main point here is that the Gaussian process emulator gives you an error which is 0 on the training dataset, and the training dataset here is identified by these black dots. So with the Gaussian process emulator, you have a 0 error on the training dataset, and you have this sort of lobs in the uncertainty, which grows when you are farther away from training dataset. Whereas with other nonlinear interpolation methods, what you see is that the outcome of the interpolation is something that, in average, minimizes the error between the function, the interpolator and the observed data, but it can very well happen, as it does in this case, that you don't have 0 error anywhere because you are interpolating in average. Whereas as I said multiple times already, the Gaussian process gives you 0 error on the training dataset, and then the error changes smoothly between 2 consecutive points. And this is an example of a pipeline that combines the use of Gaussian process emulators and the use of sensitivity analysis for the 2 different applications that we will be focusing on in the rest of this talk. At the very beginning here, you have the essentially the definition of your model, where you define the input parameter and compute all the associated uncertainties, and you run the model on the input parameter space. Now, once you run the model on the input parameter space, you can train the Gaussian process emulator to emulate the output distribution, and you can then perform the sensitivity analysis for decomposing the variance, and the variance decomposition, we will see how it works, how it is associated with the task of identifying biomarkers. Or you can rank the parameters and perform a model reduction. But a very important result that is shown in this slide here is that it's been observed that you actually, for a model with n input parameter space, you typically need in the order of n simulations for training the emulator. And this is very important because if you remember, I said that you need 1000 times n model evaluation for performing the sensitivity analysis, but you only need n for training the emulator. So you could actually run only n simulation and then use the results to train the emulator, and with the emulator generate the remaining points that are needed for the sensitivity analysis. And this gives a significant allows to save a significant amount of time and computational resources, especially when dealing with very large problems. Now this is the end of the introductory part. And now I will go on presenting two applications we have been working on here in Sheffield with the use of Gaussian Process Emulators. The first application is in the cardiovascular domain, and here we have used the Gaussian Process Emulator for identifying biomarkers for ischemic stroke. And these biomarkers are can be easily used in the clinics, as we will see. So we're talking about ischemic stroke, so we focus our attention on the brain. Now, the brain is a highly vascularized region of the body, and actually is the organ that receives the most amount of blood. And inside of the brain, there are thousands of vessels. But amongst all these vessels, a structure of particular interest is the circle of Willis, which is a ring of vessels just at the center of the brain, which has the responsibility of distributing blood to various parts of the brain, and it also provides alternative paths for the blood in case a vessel of the circle of Willis itself is occluded. So you have, what happens is that you have blood coming from here or from these two vessels, and then the blood gets to this point here, which is the circle of Willis, and gets redistributed along these directions. But what can happen, and unfortunately happens very often, is that one vessel can be occluded by a clot of blood. A clot of blood is called, and this event of occluding a vessel with a clot of blood is called an ischemic stroke. And so one of the vessels that that gets occluded more often is the middle cerebral artery, which is this large artery here, which is responsible for providing blood to a large part of the brain. And of course, if you have an occlusion here, what will happen is that all the tissues downstream will suffer because of the reduced perfusion. And in this case, you can already see it by the topology of the network here. In this case, the vessels of the circle of Willis are of little help in bypassing the occlusions and bringing blood to the occluded, to the region that is suffering. But fortunately, the brain, besides the circle of Willis, which is the first, the primary collateral path, it also presents a number of small vessels, which are called the leptomeningeal anastomosis, or LMA, which are responsible for providing blood to this region when there is a major occlusion here. And you can see the leptomeningeal anastomosis here in this picture. So the leptomeningeal anastomosis are very small vessels which connect the anterior part of the brain, is this one, or the posterior part of the brain, which is this one, with the middle one, which is this one. And they this connection is bilateral, so it's bilateral. So the blood can flow either from ear to ear or in the other direction. What happens is that when the MCA is occluded, there is a flow redistribution inside of the brain, and this changes the pressure in the vessels, and this change of pressure induces a pressure gradient at the extremities of the leptomeningeal anastomosis, which again they are here but not visible. And what this pressure gradient does essentially is pushes the blood from these regions which are perfused to these regions which are currently suffering because of low perfusion. And in this way, it's possible for these regions that are suffering to not die immediately. So essentially, the leptomeningeal anastomosis buy time to the patient who's suffering from a stroke. And as you can imagine, more extensive networks of leptomeningeal anastomosis provide better alternative perfusion routes. And in fact, it's been observed that patients with good leptomeningeal anastomosis tend to recover better after a stroke. So you can imagine that knowing how effective this network of small vessel is would be very important in the for stroke management. So if doctor knew how well or not well this network is working, they could have an idea of what is the level of perfusion here and what is the status of the patients. Now, the problem is that the leptomeningeal anastomosis cannot usually be observed directly. They can be observed only in limited circumstances and only during stroke, where, of course, time is very important. But even though if they cannot be observed directly, they offer indirect signs of their presence, and these indirect signs can be monitored. As I said before, after a stroke, there is a redistribution of the blood inside of the brain, and this redistribution changes the velocity in some of the arteries of the brain. So if we could measure the velocity, we could try to infer what is happening inside of the brain itself. And actually, it is possible to measure the brain velocity, the blood velocity, and this can be measured by means of the Transcranial Doppler ultrasound, or TCD. TCD is a device that sends sound waves into the skull of a patient and estimate the blood velocity in the underlying vessel by measuring the frequency shift of the reflected wave. So basically, you probe the head of the patient, the device sends some waves in, the waves are reflected by the red cell flowing inside of the arteries, come back, you measure the frequency shift, estimate the velocity. Now, this transcranial Doppler ultrasound is currently used for stroke diagnosis, and it has the advantages of being relatively inexpensive and fast to use. The drawback is that it only measures the velocities, and it only measures the velocities in proximal locations of the brain. And it does not inform on what is the level of distal perfusion downstream the occlusion. And this is actually what doctors are interested to know, because they want to estimate the status of the patient. But this leads directly to our research question, which is it possible to use TCD measurements to identify biomarkers for distal perfusion following a stroke in the middle cerebral artery. Now we will try to answer this question using a combination of mechanistic model and mechanistic modeling and statistical modeling. The mechanistic model will be the development of a computational model of the brain circulation, and with the statistical part we will describe the brain circulation in a large population of stroke patients. Sorry. Okay, so the mechanistic part involves the development of a one-dimensional model of the brain circulation, which contains the main arteries of the brain, this one red, blue, and yellow. And it also includes the leptomeningeal anastomosis, which are these small vessels here in green. In the one-dimensional setting, we model vessels. The vessels are straight, long tubes that can only deform in the radial direction, and they deform to accommodate the variable flow regimes that are characteristic of a cardiac cycle. The model here is developed using the software of OpenBF, which is a software that we have developed here in Sheffield and is available open source on GitHub. And this model takes as input a number of parameters, but in particular, it needs the radii of the arteries in the network. And given this, it's able to predict the velocity, flow rate, and pressure and perfusion in all the points of the network. So the mechanistic part forms the basis for our statistical model. Now we want to describe a large population of stroke patients. So we needed to generate a large population of stroke patients, of course, and we generated 110,000 virtual subjects. But of course, we couldn't simulate all of them using OpenBF because each simulation takes 10 minutes to run, and it would be really unfeasible to explicitly simulate all of the patients. So what we did, we trained an emulator on a smaller data set and generated all these points for generated all these virtual subjects. And in this case, the input of the emulator were the radii of the network, so essentially it's geometry. And we also included a representation of the uncertainty that we have on those radii, because as you can imagine, they are not known exactly. And given this distribution of radii, the emulator was able to predict the perfusion in the distal part of the network, and this is what the doctor would be interested in knowing, but they cannot know because it's not measurable. And then the emulator also predicted the velocity in 6 different points, and these are velocities that are actually measured in the clinics, and doctors use this velocity. So the emulator outputs a combination of things that are either measurable or non-measurable. And once we had the trained emulator, we looked for biomarkers using the sensitivity analysis. So we, on the right here, you can see the output of the sensitivity analysis, and these results are shown as a heat map. So on the x-axis here, you have the input of the model, and on the y-axis, you have the output of the model, and darker color signifies an higher influence by the input on the corresponding output. And remember, what we are interested in is the distal perfusion, which is not measurable, and is this variable here. And if you look at the variables that affects it, you see that essentially it's only determined by the radius of the leptomeningeal anastomosis. So ideally, if we look for a biomarker, we're looking for a variable that behaves in the same way as the average perfusion and is affected by the same set of parameters. So we looked for a function of the observable velocities that had the same signatures in terms of dependency. Unfortunately, we couldn't identify a variable that behaved exactly in the same way, but we identified four different biomarkers, four different functions that had a fair dependency on the radius of the leptomeningeal anastomosis as the average perfusion, but there was also some contribution from other parameters of the model. And after having identified the biomarkers, we generated another large population of subjects to test how the biomarker behaved. And again, we generated all of these subjects using the emulator. And what we found is that the biomarkers could not predict exactly the level of perfusion, and this is a consequence of the fact that there is dependency also from other parameters of the model. But the biomarker could be used to identify thresholds that inform on the probability that the level of perfusion is above or below a certain threshold of clinical interest. So this would help the clinical decision making in emergency situation, because from the value of the biomarker, the doctors could at least have a rough idea of what are the distal conditions. So this concludes the cardiovascular application. I will now dive straight into the musculoskeletal one. And this is a study that, again, we are performing. It's currently being performed here in Sheffield and concerns the use of Gaussian process emulators for doing model reduction of personalized musculoskeletal models. I will start with a brief definition of what a musculoskeletal model is. It is a mathematical representation of human locomotion. Now, as you can imagine, it needs a musculoskeletal model needs a combination of input and provides a number of different outputs. And now I will only give you a high-level description of the modeling pipeline. If you're interested, you can send me an email and I can provide you with more details. So the input of the musculoskeletal model are the trajectories of the body segments during a locomotion task, and these are measured using a set of cameras. And then you also need the forces that the body exchanges with the ground, and you need a description of the geometry of the anatomy of the patient. Once you have these three data, you can run the musculoskeletal model, and this computes a series of intermediate variables that are needed to get to the end, where you have the forces that are generated by the muscle to produce the observed motion task. And finally, and this is the important thing, you can compute the forces that are acting in the joints of the lower limb. And the reason behind the interest in this, in knowing the joint reaction forces, is because these forces are often associated with the onset and progression of pathologies of the musculoskeletal system. So, for example, osteoarthritis, it's highly affected by the magnitude of the force acting on the joint. Now, the anatomy of the body can be provided with various levels of personalization. You could either use generic atlases where you don't specify too much if you use an atlas, the anatomical detail will not be matched to the subject that you are examining, but you could also perform MRI scans of the subjects and perform a segmentation of all the bones and all the muscles, and this will give you very precise estimates on what are the inertial properties of the lower limb, and also what are the parameters associated with the muscle that describe how the muscle generates force. And if you use this sort of pipeline, you will end up with a model that is extremely representative of the patients that you are studying. Now, a typical model usually contains 23 muscles per leg, and they are split into 29 muscle lines, so roughly you have 60 parameters. Musculoskeletal simulations are usually quite fast to run, so they take 5 minutes, but before you can get to the simulation, you have to segment the muscle, and a skilled and experienced operator can need up to 20 hours to perform the segmentation of the lower limb. So the question comes naturally, could we only segment the important muscles and focus our attention on the important muscles and run and consider the other muscle with a minor level of personalization? Of course, you have to answer how do you rank muscles, and as you can imagine, you can rank them using sensitivity analysis. And the aim of this study here is to develop a reduced model of the musculoskeletal system, where we only personalize a limited number of muscles, but we want, of course, this reduced model to be as accurate as the full original one. And it's done using a combination, again, of mechanistic MSK simulation to generate the input dataset, and then the emulator and the sensitivity. We had available a dataset from 11 women who performed a variety of motion tasks at various speed, and the motion was recorded with the cameras in the gait lab, and they also performed they also underwent an MRI scan of the lower limb. So we segmented all the muscles of all the women and extracted the statistical distribution for all the muscles. And once we had the statistical distribution for all the muscles, we were able to draw 200 virtual patients from this distribution, and these patients were representative of this population and could be used to could be used as input for the musculoskeletal simulation. So we simulated 200 different women, virtual women, and computed the hip, knee, and ankle joint reaction forces during gait. The question now is, why do we want to use an emulator if the model runs only 5 minutes? Is because if you have 60 parameters, you still need to perform in the order of 60,000 simulation, and at 5 minutes per simulation, this amounts to 200 days, roughly. But if you use an emulator, you only have to perform order of n simulation. This is 5 hours, and then a little bit more for the Sobol indices. So again, significant reductions in time. And so we ran all of the simulations, all of the musculoskeletal simulation, the 200 one, and we split the results from the 200 into a training data set and a validation data set. And we trained the emulator on a small data set of 50 simulation, and the emulator accepted as input the muscle parameters of the subject and was able to predict the entire time-dependent joint reaction force during the gait. So unlike the cardiovascular application, where we use the emulator to produce single values, in this case we are actually emulating entire time-dependent functions. And as you can see here, this is the example for the joint of only one virtual subject, but the emulator was able to predict joint reaction forces that matched closely what was produced by the simulator. So we were confident that the emulator was working very well. And we used the emulator to generate I think it was 500,000 virtual subjects to run the sensitivity analysis. You see the outcome here of the sensitivity analysis. For the 3 joints, you have here on the x-axis you have the gait cycle, and on the y-axis you have the muscle of the joint, of the leg. Sorry, we identified which muscles had the higher contribution to the joint contact force during the gait cycle, and we were able to identify 5 muscles that had a higher influence on the determination of the joint contact forces. So these muscles were then used for performing model reduction, and we built a model where we personalized only 5 muscles of the 29 in the leg, while the remaining muscles were the values for the remaining muscle was chosen from the average of the population. And then we simulated the response of the reduced model, which is here you can see it with the one with the gray circles, and we also simulated the response of the full model, which you can see with the black solid line. And as you can see, there is a quite satisfactory match between the two. And this means that the reduced model, where you only personalized 5 muscles instead of 29, is actually able to explain much of the variability and to match the behavior of the full model. So this concludes the musculoskeletal application. The take-home message here is that Gaussian process emulators are effective tools for performing sensitivity analysis of complex computational models in different domains and different features of the output. So scalar values or time-dependent functions, they behave well in all of these cases. Want, of course, to acknowledge and thank my funding, ComBiomed, which is hosting us, and Sano, and also the Multisync project for the musculoskeletal application, and all of you for your attention.
Brilliant. Thank you very much, Ivan. Just a quick reminder for the audience that if you do have any questions, then please go ahead and write them in the questions tabs, and that way I can read them out for you. So just whilst we're waiting for people to gather their thoughts, I've actually got a few questions of my own for you, Ivan, if that's okay. So you just gave a really striking example in which that musculoskeletal model can be reduced quite dramatically. So is there any identifiable structure both in that given example, but also more generally, that would hint at a problem being more or less amenable to model reduction via Gaussian process emulators?
Well, yes, I would say that in this case, we were able to identify these 5 muscles. I mean, thanks to the Gaussian process, but the Gaussian process was a tool for doing that. I think it was possible to identify these 5 muscles because of the physiology of the musculoskeletal system. So they emerge quite naturally as the more influential in this case. So I think that Gaussian process emulators are a great tool for emulating the systems, but what really determines whether it's possible to produce a good reduced model is the inner structure of the model itself. So if of these 29 muscles, all of 29 were equally important, well, it would not have been possible to develop a reduced model.
Sure.
Yes. I think what really drives what can be done with the emulator is the structure of the underlying system that you're studying.
Okay, sure, I understand. So had all 29 of those muscles been significant, I suppose that would have been identified by the GPE?
Yes, yeah, well,
Sure.
yes, by the sensitivity analysis on the points that were generated by the GPE, yes.
Okay, okay,
You would have seen, like, for example, here, you see there are, for example, the gluteus medium here, this is quite darker than the surrounding muscle.
Sure.
If all of them were more like equally influent, everything would have been a bit darker, but you would not have seen these peaks here. It would have been a blend of color, essentially, where the influence was equally split between the 29 muscles.
Okay, yeah, of course, I'm with you. So have I understood correctly that musculoskeletal model example, that was performed using real patient data, right?
Yeah.
So do you have any other examples of where GPEs have been practically validated like that in the clinical setting?
Not many, actually, because it's quite hard to bring these technologies to the actual clinical practice. It really depends on the sort of application, on how critical it is to the patients. But just these days, I was reading about an application where they have used the Gaussian Process Emulator to predict the progression of Alzheimer's disease in patients. So they used an input for the emulator, a number of clinical data that were collected throughout the year, or longitudinal data, and trained the emulator to predict the score that measured the progression of the disease. And this was, I think it wasn't a clinically adopted study, but again, it used clinical data and was practically validated on this data, although I don't think it was directly used in the hospital. So this is an example of application of the GPE on real patient data.
Well, yeah, no, I see. Is there any particular domain that you can kind of foresee GPEs being particularly beneficial to?
Well, I can think of my domain, the cardiovascular. I mostly work on the cardiovascular domain, and I think they can really do a great job in working with uncertainties, because I'm thinking of the brain. You don't really know you don't have often the data on the brain. I mean, you don't. Of course we do have generic atlases, but when you have a patient in front of you, that patient doesn't necessarily match what's in the atlas, and you could use the emulator to try to skew the prediction from the atlas in a particular direction. Maybe you personalize a little bit of the parameters, but with the emulator you can actually study the influence of all these parameters. And for example, you say, I need to know just this particular rate, and this is actually a part of the study that I have not presented here for the cardiovascular application. What we saw is that you needed to measure with a reasonable accuracy only 2 or 3 vessels, and you need to know the radius only of 2 or 3 vessels, and this is reasonably easy to do in the clinics. And with only information on 3 vessels, you could reduce the uncertainty on the results in distal part of the network where you have reduced information. And the GPE actually told us this.
I see. Thank you. Yes, no, you have done. Thank you very much for that. Now I can actually see we've got quite a few questions coming through from the audience here, which is good to see. So Roel Myberg says, thank you for a great talk. How does GPE compare to other meta-modeling approaches such as polynomial chaos expansions?
In all honesty, I'm afraid I cannot answer specifically on this. I know that they are No, I'm sorry. I don't know the specific answer, the detailed answer to this, and I don't. I can. If you send me an email, I can come back to you later.
That's fine. They have a
but
follow-up
I'm not
question as
sure
well.
I'm not sure to reply clearly, and I'm sorry about that.
That's fine. There's a follow-up question from them as well. So how do you check if your GPE is, in a sense, good enough in emulating your simulator? Do you have to perform some sort of leave one out error tests?
Yeah, you can do the cross validation, or essentially what we did here is we had the training dataset and the validation dataset, and we split them. And what we did is essentially you train the emulator on the validation on the training dataset, and then you test it on the emulation on the validation and compute the associated error. And if the error is above a certain threshold, you increase the size of the training dataset and then perform the validation again. And if the error is still above the threshold, you further increase, and so on until you get to convergence, and your error is below a threshold that you decide at the beginning. And of course, the split of training, validation, and testing can be done with leave one out or other cross validation techniques.
Great, thank you. I've also got two questions from John McCulloch. So first of all, how reusable are the GP models, such as for the cardiovascular case? Would a different occlusion location require full retraining?
Yes, quite clearly yes, because the topology of the network changes dramatically. So all the redistribution patterns are quite different. So we have if we have the occlusion here, the blood is not entering this region and is going through ear and through ear. But if you occlude a different vessel, then you are studying a completely different problem, and you have to retrain the network. Yes, at least that's what we have seen. Maybe if you have the occlusion in the same vessel, you can move it further away or closer to you by tuning the length parameter, for example. But if the vessel is if the occlusion is in another vessel, yeah, you have to train again.
Okay, right. And then the second question from John. So a back of the envelope calculations might suggest that parallel deployment of your full model on a large-ish HPC could potentially reduce the simulation time below the GP training time. Do you have any comments on
that? If you do have access to to such a machine, yes, of course. We actually performed many of the simulation in parallel. We could parallelize and run only 300 of them at the same time, which was still a significant reduction. You need to have access to such a machine, John. I do agree with you that if you do, of course, it's very beneficial. But then every single time you have to run another model, you have to, every single time you want to evaluate another point, you have to run the model again. And for example, I don't know, if your model takes 10 hours to run and you are able to run an infinite number of parallel simulation at the same time, that's fine. But then if you want to simulate another point, you will have to wait 10 hours because you don't have the emulator trained at that point. So it's always a trade-off between what you can do and what you want to do, I think.
Right, and on the topic of high-performance computing, I understand that GPEs are implemented within the VECMA toolkit. So what are the challenges associated with deploying this GPE approach at the exascale?
Well, one is one challenge I see is related to what I was saying here. So that, I mean, exascale simulation take very long time, and you will have to run have to run a very large number of simulation, and it may not be always possible to parallelize them. So that's an issue. For example, just the ability of producing the data that are needed for the GPE can be difficult. And also there is an issue, I think, about the capability of the emulator of scaling up. So exascale simulation are very large, typically in size. They have lots of input, lots of output, and the GPE sometimes struggle when the input parameter and the output parameter space grows too much, and you can run into troubles for the training. And of course, you have to be, I mean, if you have an exascale model and you select a limited number of points and always assuming that you're able to run all the simulations required, then fine, the GPE will work very well. But if you, for example, have an exascale model of the entire circulation, and you want to have an emulator which is very detailed in emulating the exascale simulation, this might be an issue. You could try to overcome it by some statistical modeling or data reduction, where you define bases, and so rather than emulating the entire solution, you only emulate the coordinates with respect to a certain base. But this is just a speculation that I have. We haven't tried doing this.
Sure, I understand. So I'm not seeing any questions coming through. I'll just ask you maybe one or two more, and if there are any last minute questions from the audience, then I can ask those. So in the training phase for your GPE model, is there a danger of overfitting the model? And if so, what sorts of precautions might be taken to circumvent that?
Yes, it can happen. You can obviously always, you should always check the convergence of the model and how it behaves on the validation dataset, and you could work on the relative ratio of the training versus the validation dataset. This could be an option. You could change the kernel, for example, because maybe one kernel can be more exposed to the risk of overfitting for a certain dataset structure, whereas another one maybe is better. Or you could add some noise artificially to the kernel itself, and this could, in theory, help a little bit.
Oh, okay, yeah, sure. And one more question from me. Would you be able to describe a little bit the difference between Gaussian process and multi-layer perceptron networks?
Yeah, so with the Gaussian process simulators, in theory, we could optimize them exactly, just performing the derivative of the emulators. Whereas with it's my understanding that with the multi-layer perception, well, you can't actually write the solution of the optimization and find all the parameters in a easy closed form, but this requires the back propagation and the numerical optimization. Then, as I was saying, the GPEs are excellent on small, medium datasets, but they can struggle when scaling up to large datasets, whereas the neural network tend not to have this problem. And the Gaussian process work very well on smooth data because of the underlying assumption of smoothness. But when the data are less regular, the accuracy of the emulation can go down. Yeah, I would say these are the well. Thing is that with the Gaussian process, you can also naturally include a representation of the noise, whereas I'm not sure you can do this, at least with rudimental network. With the MLP, for example, I'm not sure if you use more complex and defined network, but with the Gaussian process, it comes very naturally. You just add a noise term to the kernel, and this is something that, as I said, can also help with reducing the risk of overfitting.
Right, I'm with you. Yes, I see. I haven't had any more questions come through in that time, so I think this would probably be a good point to draw the session to a close. So thank you once again, Ivan, for giving us your time today to speak about Gaussian process emulators. I certainly got a lot out of it. Just one final reminder that this e-seminar will be available on the CompBiomed YouTube channel and website, which is accessible via the link shown on screen currently. So finally, I just want to thank everyone in the audience for attending, and I hope that you all enjoy the rest of your day. Thank you.
Thank you very much to you, team, and to everybody who attended.